Hello, welcome back to the morning session. I hope you enjoyed the poster session. And now we'll start with Professor Werner Treptow, who is going to talk about allosteric modulation of membrane proteins by small ligands, application to ion channels and general anesthetics. Hello.
Okay. Hello, everybody. So, can you hear me?
Yes, we can.
Okay. So, I will share my screen
please.
now. So, can you see?
Yes. Now, yes.
Okay.
Thank you.
Okay. So, thank you. So, yeah. So, I would like to start by thanking all the organizers for the invitation. Definitely, it is a pleasure for me to be here today contributing this meeting. Unfortunately, we're not all together in Rome, but I hope everybody is safe and healthy. And today, my talk is on the allosteric modulation of membrane proteins by small ligands. And by allosteric modulation, I precisely mean that a ligand may bind a protein to shift its conformational equilibrium between two or more conformational states. But for the case of a small low affinity ligand, that modulation can be very complicated because the binding process is highly complex. As shown here, the three-dimensional densities of the ligand on the surface of the protein can be spread over multiple sites. Each site can be occupied by two or more ligands in such a way that the binding is necessarily concentration dependent and takes place under complex saturation conditions. So what I would like to do today, first I would like to elaborate on a thermodynamic model that can describe this kind of complex situation here. And second, I would like to apply that model on the investigation of ion channels and anesthetics, which I believe it is an important problem ranging from biophysics to neurosciences. As you know, membrane proteins can respond to a variety of stimuli in nature in such a way that we may record experimentally their response probability as a function of a number of variables in the system, and changes in the surface tension of the membrane or transmembrane voltage or the pH. So if the membrane protein is further modulated by ligands, so then ligands may induce a shift in that probability, as shown here. And that shift may be rationalized in terms of a thermodynamical cycle in which the ligand may bind the protein and shift its conformational equilibrium between two states. So let's say closed or open, or resting and activated, as shown here. So first, what I would like to do, I would like to solve this cycle very briefly here. And then we apply the cycle on our investigation. By solving that cycle, we start by treating the protein as an open molecular system. So the protein is embedded in a large environment, membrane water environment, as shown here. The protein and the membrane water environment forms what is called an extended system, which is coupled to an external device that is used to stimulate the system at a constant value chi. The environment is further assumed to contain a large number of ligands in such a way that the environment provides with a reservoir volume for the ligands. And because boundaries are open, so ligands may migrate from the reservoir into the protein sites to bind the protein in a reversible manner. The free energy of this whole construction here may be defined for a specific conformation of the protein S, for the position of the ligands in the system R, in terms of the microscopic potential energy of this extended system, plus the energy of the external device. So chi bar is what is called intensive extensive conjugates, And for the case of membrane proteins, this typically corresponds to an applied transmembrane voltage and its conjugate charge displacement, or even an applied surface tension and its conjugate lateral area, or even a chemical potential of protons and the conjugate number of bound hydrogens. The definition of that free energy is important because, well, we may then define the free energy difference between conformations of the protein, X and Y, when the protein is stimulated at a constant value chi. And we may define that free energy difference for the specific situation where all the ligands are in the reservoir volume, meaning that they are not bound to the protein. Further expansion of that free energy difference in terms of two components, the intrinsic conformation free energy of the protein, which is the free energy between conformational states in the absence of any stimulus, plus an excess free energy that appears in the system when you stimulate it. So from this expansion, we may finally write down the probability of the protein to be in a specific conformation Y, free of ligands, and stimulated by chi in the form of this familiar two-state Boltzmann equation, which is really very popular among experimentalists because it may be quantified in terms of experimental parameters obtained directly from the response probability of the protein. Okay, once this probability is known, solution of the cycle proceeds by solving the binding reaction to a specific conformation of the protein in the absence of any stimulus. And because we are interested on the case of a low affinity ligand, probably we wanted to solve a generalized reaction where one or more ligands may bind the protein over multiple sites. So as largely discussed in the literature, solution of that reaction involves the quantification of the bind constant, which account for the free energy difference of transferring ligands from the reservoir into the protein sites. And of course, that bind constant must take into account the fact that ligands are indistinguishable and that they have a specific concentration in the environment. Knowledge of that bind constant is very important here because from here we may quantify the occupancy probability of the protein in a specific conformation. So it's occupancy by a specific number of ligands. And therefore, we may quantify any ensemble average property of the system in a concentration-dependent manner. So if you put these two pieces of the cycle together, we may then finally write down the probability of the protein to be in a specific conformation Y and stimulated by chi, in the form of the Boltzmann equation as before, but now taking consideration the ratio between these two grand canonical partition functions that accumulates or describes the affinity of the ligand across every possible occupancy state of the protein in a specific conformation. So in the present form, that probability is a generalization of the classic Monod, Wyman, and Changeux allosteric model for proteins. And its meaning is quite straightforward. So if the ratio is smaller than 1, it means that the ligand binds preferentially the Y state, and by doing so, it shifts the response probability of the protein leftward. Otherwise, it shifts rightward. Okay, so we are done. We think this equation is quite general. It can be used for a variety of biochemical studies of membrane proteins and ligands. And where we think this can be very important is on the study of ion channels and general anesthetics. So as you know, general anesthetics are small uncharged molecules. And here I show you typical exemplars, including propofol, halothane, and sevoflurane. And despite efforts reached back over a century, their action in general anesthesia remains largely unknown. The current view is that ion channels in the brain are implicated, and key players have been identified so far, including the inotropic neurotransmitter receptors, voltage-gated ion channels, and non-gated ion channels. So somehow, general anesthetics are expected to impact the ion channel function, to change their conductance state, and therefore the overall excitability in the brain. So an important set of experimental and theoretical finds support that the anesthetic action is mediated by the membrane by means of physical or biochemical processes. For instance, the anesthetic is expected to bind the membrane and to change the lateral pressure applied on the ion channel. Or even the anesthetic is expected to bind and to trigger a cascade of biochemical reactions involving lipids or second messengers that change the function of the ion channel. And alternative to that possibility, another important set of studies, including structural studies, relied on crystallization or MD simulations, points to another possibility in which the ligand is expected to bind the ion channel directly. Intrinsic in that possibility is the assumption that the ligand is expected to bind, and more importantly, is expected to bind in a conformation-dependent way. And this is precisely the kind of hypothesis we would like to be investigating here in the context of the voltage-gated potassium channel Cave 1.2 and the general anesthetic CO41. Cave 1.2 is largely expressed in the brain, and SIVO is commonly used in hospitals. And over the recent years, Covarrubias and co-workers showed that at 1 millimolar concentration, SIVO changed the open probability of the channel in Herc cells. So what we would like to be doing from here is to solve or to investigate that macroscopic shift here in terms of this minimalistic molecular thermodynamical cycle, where SIVO is expected to bind the resting close and activate the open conformation of the channel. And our particular interest on these states of the protein derives from the fact that over the years, we have accumulated structural information on these states of the channel. Very briefly, we have simulated the channel embedded in a phospholipid bilayer. So the simulations started from the X-ray structure of Cave 1.2, which was published by McKenna and co-workers in 2005. Embedded in the membrane and under applied transmembrane voltages, the system was deactivated. And in these simulations, we have considered this protocol, what is called the charge and balance protocol. And every time a protein charge is displaced across the transmembrane field, we have to rebalance the electrolytes to keep the applied voltage constant over simulation time, as shown here, in a such way that in this kind of simulations, fluctuations of the applied voltage store direct information on the gating charge being transported by the protein. And the gating charge was found to change drastically over simulation time in these kind of studies as a result of sliding down motion of the S4 helix. So again, S4 helix is this important helix in the voltage sensor. It's positively charged. And during the simulation, it moves downward and it pushes the S4S5 linker to close the pore. So the S4S5 linker is this linker in green here, and is an important piece of the machinery coupling voltage sensor and the pore. What is important to say here is that this closed structure is consistent with most of the experimental finds for this specific channel. In particular, it accounts for the transport of 13 elementary charges, which is in good agreement with the experimental measure for this gating charge for this specific channel, in a such a way that we believe this structure is representative of the resting down state of the channel. And indeed, that structure is very similar to other structural models proposed for the resting closed state of the channel. Some models proposed by independent groups following independent strategies in a such way that today in the literature, there is this consensual view that Cave 1 is activated by voltage following a cycle between these kind of molecular states. So according to these findings, we have relied on these two molecular structures to solve that cycle. And we start our investigation by looking at the interaction of SIVO on the open pore structure of cave 1.2. We have investigated that interaction by means of docking and free energy calculations. And very briefly, we start by simulating the channel in a phospholipid bilayer in order to generate a large ensemble of conformations as shown here. Then SIVO was docked against that flexible ensemble to solve interactions of the ligand at the S4-S5 linker, at the S5-S6 interface, and at the external face of the channel near the selectivity filter. Because we were interested on saturation, complex saturation conditions, SIVO was then redocked against this first generation, first ensemble of docking solutions to solve double occupancies at the S4-S5 linker, at the S5-S6 interface, but not on the external face of the channel. Here are exemplars of the molecular solutions at the S4-S5 linker for single and double occupancies. Then the affinity of the ligand, the bind affinity of the ligand to each of these sites was evaluated by means of free energy perturbation calculations by taking consideration the whole channel with explicit water and membrane. And in this table, we summarize the results we found from FAP calculations, showing bind affinities for single and double occupancies of the bind sites. So the bind affinities are in the range of millimolar concentration, meaning that the ligand may bind the protein, but bind is necessarily low affinity. In a such way that, well, from these numbers, it appears that SIVO may interact with the open pore. Accordingly, we have relied on the fact that bind sites are sparse on the surface of the channel, and we relied on these individual site contributions here to compute the aggregate bind affinity. So aggregate bind affinities for any given occupancy state was computed by assuming that bind sites are independent, and we consider saturation conditions up to 2. And from the binding constants solved in this way, we then calculated the occupancy probabilities to the open pore. And here we show you the results. So this is the probability of the protein to be occupied by a given number of ligands, regardless their site distribution. And it's clear that as you increase concentration of the ligand in the environment, you start populating more and more the protein. That population is, however, highly site-dependent. There is a clear preference for the S4-S5 linker, followed by the S5-S6 interface, and a much less important interaction at the external face of the channel. Visualization of these results in 3 dimensions show very clear that the SIVO may bind the protein at the S4-S5 linker at 1 millimolar concentration. And as you increase concentration in the bulk, you start populating the S4, the S5-S6 interface as shown here. So the overall conclusion from these calculations is that SIVO may bind KV1 in a concentration-dependent manner, binding the S4-S5 linker and the S5-S6 interface. These results are consistent with past findings by Koval-Rubias and co-workers, showing that alanine replacements at the S4-S5 linker drastically impact the energetics of anesthetic interaction with the channel. And more importantly, it's highly consistent with Eckenhoff and co-workers, showing very independently that SIVO can protect the S4-S5 linker and the S5-S6 interface in photolabeling experiments. Protection is higher at the S4-S5 linker, which is pretty much consistent with our calculations. Later on, we extended our calculations to investigate the interaction of the ligand with the entire transmembrane domain of the protein. And more importantly, we extend our calculations to look at the 2 conformational states of the protein. So docking calculations were performed as described before. So we could find the same bind sites as before at the linker at the S5-S6 interface, but other sites were identified within the voltage sensor, and more important, at the S4, S5 interface, as shown here. Binding constants were then quantified from free energy perturbation calculations, as described before, and the probabilities were analyzed. So the three-dimensional densities as a function of the concentration shows clear difference between the open and closed states. Indeed, quantification of the occupancies as a function of the concentration shows an excess number of bound ligands to the open state. So the excess is 3 molecules in average at 100 millimolar concentration. So there is a concentration, there is a conformation-dependent effect on the binding, and the impact of that concentration dependence on the energetics of the protein was investigated by quantifying the open probability of the channel. So that open probability was quantified by taking consideration the specific conjugate for the channel, which is the gating charge and the voltage. And here I show you the results. So these are open probabilities versus voltage. I show here the experimental results by Covarrubias and co-workers in black and in blue. And in red, in green, and orange are our structural calculations, showing that we may recapitulate the experimental shift in the range of 10 millimolar concentration, which appears to be in qualitative agreement with the experimental finding. Further decomposition of that probability in terms of site contributions, as shown here, reveals a complex, non-trivial interplay between sites. At 100 millimolar concentration, the S5, S6 interface actually stabilized the closed conformation, but that effect is largely dominated by strong stabilization of the open state by the S4, S5 interface and by the S4, S5 linker, as shown here. So at this point, these calculations appears to rationalize the experimental findings in terms of the direct bind events, where the S4 pore interface and the linker appears to play a major role in the stabilization of the open state. So these results are fine, they are interesting, but it's true that they may suffer from imperfections from our calculations. So for instance, imperfections on the energy function in docking calculations, or limitations of the sampling of the bound state in the free energy perturbation. So accordingly, we decide to investigate the dependence of these results with the calculations. So we try to follow a different strategy for that goal. We performed flooding MD simulations. So we flooded the channel embedded in a phospholipid bilayer with an excess of ligand molecules. The ligands were simulated at a high concentration, 150 millimolar concentration, which was found to be high enough to sample the bound state at reasonable time scales, but low enough to avoid aggregation of the ligand in solution. And from these simulations, we see very clearly that the ligand migrates from water into the membrane and then binds the protein in a reversible manner. So the simulations were performed for the two conformational states of the protein. And here I show you the analysis of the average number of bound ligands as a function of a given binding cutoff distance. So let's say 5 angstroms. And there is a clear excess of bound molecules to the open state of the protein. Among these 14 molecules, 8 molecules are bound to regions of the protein for which the root mean square deviation between the open and closed states is larger than 4, meaning that the molecule is bound to regions implicated in the gating motions of the channel. And that conformational dependence here was found despite the fact that the ligands has the very same concentration in the membrane, so in the environment. Okay, so again, the impact of that conformational dependence on the energetics of the protein was then investigated following the reconstruction of bind constant from the three-dimensional densities of the ligand recorded in the high concentration flooding MD. So that reconstruction is, however, not trivial. because at high concentration, the bind constant may become concentration dependent as well. As a result of perturbations of the protein structure, which can be very different at high concentration from the dilute situation, and as a result of perturbations of the dissolution free energy of the ligand itself, which can be very different from the ideal situation as a result of self-interactions in a high concentrated environment. By taking consideration these aspects of the problem, we believe we succeeded to develop a new formulation that hopefully will be published very, very soon. And in this formulation, we may estimate the dilute bind constant from the high concentration observed in flooding MD simulations. And according to that formulation, I show here our first estimates for the impact of the ligand on the open probability of the channel as observed in high concentration. And again, I show you here the experimental curves in black and red, which are the curves obtained by Covarrubias at 1 millimolar concentration. And in orange, green, blue, our reconstruction from high concentration. And now we have a better agreement with the experiments where the leftward shift may be recalculated between 1 to 10 millimolar concentration. So again, this appears to be in a better agreement with experiments and appears to support our docking and free energy calculations. But what about in negative control? In 2017, Covarrubias and co-workers again showed that cave 1 is sensitive to propofol. There is no clear impact on the ligand on the open probability of the channel. So then we decided to evaluate these results in the context of our formulation. We have performed flooding MD simulations as described before. The ligand was simulated at high concentration, 50 millimolar. That concentration was high enough to allow sampling of the bound state at 500 nanoseconds, but again, low enough to avoid aggregation. And that simulation was performed for the open and closed states of the channel. And again, analysis of the average number of bound ligands for any specific binding distance cutoff reveals no excess number of bound molecules to any of the conformations of the channel. These results may rationalize these experimental finds in the following sense. So propofol may bind the channel as a necessary condition. Binding is, however, very similar between the open and closed states in terms of stability. So there is no change in the average number of bound ligands, and therefore there is no perturbation of the grand canonic partition function that I explained before. In a such way that the final result is no clear impact on the open probability of the channel, therefore making sense of this result and more importantly supporting our conclusion for the case of sevoflurane. Another aspect to be controlled here, and I believe it is a quite deep one to be analyzed here, regards the assumption in the thermodynamic model that the ligand does not affect the coupling energy of the system with the external device, which means for this specific channel that the civil does not impact the gating charge being transported between the open and closed states of the channel. As I mentioned before, the gating charge is accounts for the electrical displacement of every protein charge across the transmembrane field. And by electrical displacement, I specifically mean changes in the electrical distance of the protein charge, or necessarily the coupling of the local electrostatic potential with the external voltage. So under this consideration, the condition that civil does not impact the gating charge of the protein means necessarily that it does not change the coupling of the protein with the external voltage. Accordingly, we have investigated the electrical distance profile along the gating pore axis of the voltage sensor of the protein. that investigation was performed for the voltage sensor in the open and closed state with bound and unbound silvofluorine molecule. And the computation was performed along the gating pore axis, which is the line along which the gating charges are moving during the gating process. And the level of reshaping across the transmembrane field in presence of the ligand was quantified then in the form of this parameter, MKL, which, as described in 2014, accounts for the cumulative electrical field reshaping along this gating pore axis. And the results are summarized here in this matrix. What is important to note here is that reshaping worth at most 0.1, which corresponds to little modifications of the open-close energetics in the range of thermal energy, meaning that, at least for this case, the ligand does not change the amount of gating charge being transported by the channel, therefore validating our two-state thermodynamical model, and therefore providing further support for its application on the investigation of this ion channel and that general anesthetic. Okay, so at this point, we are attempting to say that we cannot refute the participation of other molecular processes here. We cannot estimate their role. But from our structural calculations, it appears that a direct bind event may take place. For this specific model, KV1 and SIVO, we found that SIVO binds KV1 at multiple sites under saturation concentration effects. Binding of SIVO is concentration is conformation dependent and accounts for PO shifts at clinical concentrations, which was not found for propofol, for which the ion channel is not sensitive. And we found that the S4-S5 linker and the S4 pore interface are implicated. So the implication of the S4-S5 linker is not a surprise, provided its involvement in the coupling of the voltage sensor and the pore. The implication of the S4 pore interface was a surprise for us. It was unclear until last Tuesday when Pancho showed us the implication of this region on the what he called non-canonic gating mechanism. So rationalizing our findings and providing further support for the importance of this region on the stabilization of the open state. Of course, to what extent we may translate our minimalist findings here in terms of minimalist model to the great reality of an ion channel embedded in a neuronal membrane is highly questionable, and further experiments will be required to validate that assumption. Besides, it is still unclear for us the role of other conformational states on that modulation process. For instance, we are particularly interested on the activated closed conformation of the channel, in which the voltage sensors are up but the pore is closed. Still, we are very interested on non-conductive states of the selectivity filter, so how the ligand is impact the transition of the filter between conductive and non-conductive states. And for that investigation, now in the lab, we are performing flooding MD simulations. We have generalized our model to include these states, and I hopefully in another opportunity to be able to present these results in more details. For now, I would like to finish my talk by acknowledging the work of Manuel Covarrubias and co-workers. I mean, his great experimental work on ion channels and general anesthetics, especially alcohols. Students from my lab, Leonardo and Natalia, that are involved in these written works, and of course, the former members that have been largely implicated in these publications regarding ion channels and general anesthetics. So I'd like to thank all the agencies in Brazil, the University of Brasilia, for supporting our research work, and of course your attention. So I'm completely available now to try to address any questions you may have at this point. So thank you so much for your attention.
Thank you, Werner. You have some questions, so I will start.
Okay.
Armagan is asking you if these anesthetics that you mentioned during the talk are, if they are applied from only the extracellular side of the channel, from the
when they are injecting the experiments?
If they are applied, so I guess if you...
So here in this simulation, no. In our simulations, we start from the anesthetics from both sides of simulations. So we have the partition from water into the membrane.
Okay. Then we have another question from Sergey, who asked you if you can comment on if you can comment on the partition coefficient of anesthetics between water and membrane.
Yeah. So one of the aspects when you are doing the force field for these molecules, one of the aspects you have to validate is the transition between water and in the membrane. So normally, one of the quantities to be in the global optimization of the model, one of the features that must be satisfied is that partition coefficient between the water and the membrane. So over the years, especially the work by Klein and co-workers, Tarek and all the that generation that start investigating membrane at constant pressure and the studies of general anesthetics. So there is this preference for the anesthetic into the head group region of the bilayer, right? So in a such way that if you plot, for instance, the potential of mu force along this transmembrane direction, you're gonna see a well-defined minimum there.
Okay. Then there is Vincenzo, who is...
Vincenzo.
Now, yeah, I'm going to read his question. It says, great talk, Werner, very instructive. One curiosity, can the state-dependent binding of sevoflurane to Kv12 be traced to a specific binding site?
So, sorry, can you say again, please?
He says, can the state-dependent binding of sevoflurane to Kv12 be traced to a specific binding site?
Yeah, so if there is a single site that can explain apparently in the paper we have this decomposition for every site. So we have some sites that are neutral. Actually, you have a lot of sites. From the picture you can see you have a lot of sites. And in the paper we have this careful decomposition in terms of site contribution. So many sites are really not impacting the conformational dependence, and actually have a great effect deriving from these interface that I mentioned here. So from you have a larger set of sites, and when you do this decomposition analysis, actually we may rationalize the conformational dependence in terms of these three regions I mentioned here.
Okay, and then one more from Alberto, who is asking you if you please comment on the definition of concentration in experiments and then in simulation.
Yeah. So yeah, in experiments, normally you take the whole concentration, right? The entire volume of your system. In the simulation, you can do both. You can have the concentration in the simulation box, but you can also have the concentration in what is called the reservoir. So the word reservoir here, the reservoir volume here, is reserved for this specific phase of the system, which can be the membrane, which can be the water, which can be the head group of the membrane, where you have a strong localization of the ligand. So the concentrations, the description of the concentrations that we are considering here are the concentration in a reservoir, which means that is in this confined volume in a preferred phase of the system. It's not the concentration across the entire volume of the system.
Okay, so there are no more questions. So I would like to thank you again.
Okay, thank you, Gaia.
Thank you.